it's still morning for people that are in Brazil. Here, before noon, we say, midday, we say good morning. So it's still good morning for us. Good evening for you, Christer. It's really great to have you here. We are happy that you accepted the invitation. So now I'll pass the word for Magno that he will introduce you. Hi, Magno.
So good morning, everyone. And today, can you hear me? It's okay? I think it's okay. Okay. Today we have another seminar promoted by BrPROT and USP, UFMG, UFRJ. And today is my great pleasure to introduce Professor Christian Ejsing from South Denmark University. And it's a great pleasure not only because he's a great friend of us, but also because he's a great scientist. And I met Christer when he was doing his PhD at the Max Planck Institute in Dresden. He was working under the supervision of Andrei Shevchenko and Professor Kai Simons. And for those that don't know them, Andrei was a pioneer in mass spectrometry of peptides back in the 80s in Russia. And Kai Simons is just the father of the lipid graphics theory. So both of them are giants in lipid biochemistry and the lipid work. And of course, Christer is not different. During his PhD at the Max Planck Institute, he helped to develop a lipid platform for high throughput analysis of complete lipidomes from different organisms, different cell types, what is called today as shotgun lipidomics. And during that time, he published a ground-shaking paper about the complete lipidome of each cells. And I still remember it was a great achievement. And after that, he finished with the PhD with flying colors. And after that, he spent 2 years as a postdoc in Kai's lab to improve his skills on lipid metabolism, cell culture, and biochemistry in general. So after being a great analytical chemistry, he also studied a lot the system that he was proposing to study. And then in 2009, he became assistant professor directly. He went from his first postdoc at the MPI and became assistant professor at the University of South Denmark. And then in 2012, if I'm correct, please, Christer, if I'm saying something wrong, just let me know, okay? He became associate professor. Mm-hmm. Now he's a visiting group leader at the MBL since 2017. And Professor Christer works today not only with lipids and lipidomes, but also with proteomics to understand lipid metabolism. And Christer has more than 8 publications in high-profile journals. And I think with that, I'd like to thank Christer again for accepting the invitation. And now it's with you, Christer. We are looking forward for a great talk.
So thank you very much, Magno, for the kind and great overview of where I've been and what I've been up to. So that's very good. And also thanks to all the other, organizers of this nice seminar series for giving me the chance to share our work with you. So currently I have a dual affiliation, as Magno also mentioned, both at the University of Southern Denmark, but also at EMBL. Basically trying to play on 2 horses to get some great science done. And also just to make you aware that we have a postdoc position available in Denmark at the moment, which goes into the issues of doing flux analysis by mass spectrometry. So please contact me if you're interested. So today, I will start up giving you a bit of an introduction to lipids, because I believe that most of you are probably not really fully aware of all the details about them. This will include a bit of chemistry, and also one of the things that I really, really find interesting is the regulation of lipid metabolism. Then over the years, we've been trying to build what we can probably call a multiomics platform that has lipidomics, but it's not good enough to do only lipidomics. If you want to study regulation, you also need to look at the proteins, especially all of the enzymes. So I'll just briefly give you an overview to the multiomics platform, and especially the lipidomics part, which I think is most novel to you guys. And then in the second part of the talk, I will give you an update on a story that we have under review, where we basically push out the whole multiomics platform to try to study circadian regulation of lipid metabolism in mice. And then I will end up with a summary, and happy to take any questions. So before I get started, just want to say thank you to all the great people who have been in my lab and have been helping. And for the work that I showed today on the mouse studies that we've done, especially Richard, Martin, and Elena have been very important. And for a lot of the computational work that I will also give you an introduction to, Peter, Mark, and Josh have been very, crucial in that work. For the proteomics work that we're doing, we could not do that without Ole Nork or Jensen. The mouse work has been helped with Niels Fergeman and his team. And then we have had help also from computational scientists in our department called Rolf Fagerberg, and also external people. And also other people have been helping with some of the mouse work. And then I also want to thank Stephen for some of the OSID works that I will be sharing with you. And of course, all the funding. So just like to start up with this view of the central dogma in molecular biology, which we have this, that we have the DNA that encodes information that is translated into RNA, which is then basically translated into proteins. And then where I start getting interesting is then in the so-called metabolome, or more specifically under the so-called lipidome. And below this, we could then basically start looking at what are phenotypes. So that could be disease states, or whether you're fasting or you're feeding, you name it. But what I find really interesting and worthwhile considering is that if you understand phenotypes and functions, it's probably the best to be as close to them as possible. And one of the closest way you can do that is actually studying the metabolome, or in the case of the things that we're interested in, looking at the lipids and the proteins that are then making the lipids. But it might be a bit more difficult to get understanding of phenotype if you look at, for example, DNA, or you look at mRNA levels. So that's something that is quite interesting, or important for me to be on top of. So if you look at the lipids and what they're good for, what are their functions, most of you will probably be aware that they are all main components of biological membranes, such as your plasma membrane, internal membranes in your cells. But they also have crucial roles in so-called energy metabolism and transport. When you eat food, they will be taken up by your intestinal system, and then there are systems to distribute whatever you ate into white adipose tissue, muscles, the liver, or to the brain. Lipids also have signaling functions. That can be whether you're a man or you're a woman. That's the sex determination. It can also be whether you have a headache or not. And last but not least, what is typically forgotten is that they also have a crucial function in so-called mechanical and thermal insulation. And you might want to know that you're actually walking on fat every day. And if you didn't have fat pads, you would not be able to live as long as you do, because you would destroy your bones and your joints. And one example of this kind of mechanical insulation is also what you see in cancer cachexia, where people lose all their fat tissue when they have cancers. And when they lose all their fat tissue and they fall, they easily break their bones, really testifying that this mechanical function of lipids also very super crucial. Another thing that you don't really see is basically how lipids get their functions. So lipids act either by themselves, as so-called signaling molecules, or they actually work together and giving colligative properties. So imagine that you have 1 million lipid molecules. Those 1 lipid molecules will together be able to give a new kind of feature, a new type of function, which is making up membranes. A single lipid molecule does not make a membrane. Bear that in mind. Now, if you take 1 million sugar molecules like glucose, what does that make? It doesn't really make anything except that it's just dissolved in your buffer. So this is what makes lipids, in my opinion, so much more interesting and also much more, how to say, difficult to study, because they don't only act as a colligative property as a system, but they also act as individuals. And that's really important to know of when you study lipid function and lipidomics. So looking at the chemistry of lipids, what you might want to be aware of is that we have different kind of lipid categories, which is all related to their chemistry. And one of those categories are the so-called glycerophospholipids. What is characteristic is that they have a so-called glycerol 3-phosphate backbone. This molecule comes out of glycolysis. And what our metabolism can do is that it can put so-called different fatty acyl chains on these two hydroxyl groups that we see labeled here with the oxygen. One of them is the so-called SN1 position. And here you can basically do a polymerization reaction. And then you can get the very common fatty acyl that has 16 carbon atoms and no double bonds, which we annotate as 16-0. You can also have an enzyme system, or actually two, one that elongates the 16-0 to 18. And then we have a so-called desaturase that inserts a double bond. And then you get the molecule called 18-1. Now, what we cannot do, us humans, is that we cannot make this essential fatty acid called 18-2. It's a so-called omega-6 fatty acids, because you count from the back end of the C chain, and then you see there's a double bond. And this is a precursor fatty acyl that then makes other bioactive lipids, such as arachidonic acid. There's another essential fatty acid, which is called 18-3, 18 carbon atoms, 3 double bonds. This molecule we use in our body to make something called 22-6, which is a very abundant molecule in the brain, for example. Without that molecule in the brain, you basically get mentally retarded. We can do the same exercise with making different fatty acids here in the so-called SN2 position. We can also do other kind of changes to the chemistry out here in the so-called head group. We can have a proton, and then we get a so-called phosphatidic acid molecule. We can put an etanolamine molecule, 2 carbon atoms, and then primary amine. Then you get a phosphatyl etanolamine. You can methylate this nitrogen 3 times, and you get a choline. Yeah, and then you can also put glycerol and inositol, and you can do many things. So here you basically start up. Sorry. Another thing we can also do, of course, is to change the chemistry around this SN1 position. Either we have an ester, as shown here, or we can basically remove the carbonyl atom, and then we get an ether linkage, and that basically brings us into the so-called plasmalogen lipids. So with all these different factorial, how to say, groups, changing the kind of different structures, you can then end up, in theory, with more than 100,000 different lipid molecules. And this is one of the reasons why we need, of course, mass spectrometry to identify those molecules. But it's worth mentioning that it's very rarely that people, even with high-end mass spectrometers, rarely get more than 500 lipid molecules, which are called high confidence. We can get back to that in a discussion later on if you want. Now, what is particularly interesting to me is actually how are these lipids made. And to understand that, you need to look at metabolic networks. So here's a simple network we made back in the day of lipid metabolism in yeast. What I want to draw your attention to is that you have an input of glucose. You go through glycolysis, as you might know it from your basic biochemistry textbook. You get to acetyl-CoA. This you can then activate to malonyl-CoA. And now you can basically start running the so-called fatty acid synthase. It's a big enzyme complex that is basically polymerizing these C2 units, and it can make typically a C16-0 chain. This 16-0 chain can then be further transferred into the so-called elongase system. These are membrane-bound proteins now, and they can further elongate it in yeast and also in mammalians all the way up to 26 carbon atoms and no double bonds. Or we can start inserting double bonds, for example, making this 18-1 chain that I mentioned before. And now these molecules then can be combined with glycerol 3-phosphate, which I mentioned before, and then you get an intermediate called isophosphatidic acid. We can take another fatty acid chain and put it onto that molecule, and we get the so-called phosphatidic acid that I mentioned before. Now, what is worth mentioning that in yeast and mammals is that we can also get fatty acids from the outside. You are what you eat. You need your omega-6 and omega-3 fatty acids. They come into the cell. They will get activated with a CoA residue, and those fatty acids you can then also put on the glycerol 3-phosphate backbone. And now you can see in this network that there's a lot of chemical reactions. So if you love chemistry, this is of course fantastic to play with, because all of these lipid molecules are just basic chemistry that change their chemical structures when the enzymes are reacting. And what you'll see in the network is that all of these, how to say, shorthand notations with 3 letters and 1 number are all enzymes that are encoded in the genome, expressed, and these enzymes are then catalyzing the different reactions. So what you can do is that you can go from the PA molecule, you can then activate it with a so-called CDP residue, you get a CDP DAC, you can then insert a serine, an amino acid, you get a so-called PS molecule, you can do a decarboxylation either by PSD1 or PSD2, you get to this phosphatidylethanol molecule I mentioned before, and then you can metallate 3 times and you end up in this PC molecule, phosphatidylcholine. And of course, this molecule can then also be further turned over. Now, what I set out to do already in 2009 was to ask this basic question is, can we figure out how cells and tissues regulate all these metabolic networks or all these reactions at the same time? So to try to do that, one of the things you need to be aware of is actually how can that happen on a molecular level? So you can read in your textbooks that can be transcriptional regulation. It can be post-translational modifications of your proteins. For example, phosphorylation is very important. It can be enzyme stability, subcellular localization of your enzyme. It can be lipid transport, very good old allosteric mechanisms of how you basically activate an enzyme by binding an allosteric inhibitor or an activator. And then thermodynamics, that if you need a mass action, that if you don't have a precursor, you cannot get a product. So these are regulatory modules or mechanisms that you need to be aware of when you look at that. And of course, that's only one side of the coin. And the other one is what you need to be aware of is how do we actually get insights into these mechanisms? And this is what I set out in 2009, trying to get at, basically say, okay, what do we need to do this? And to do that, we delivered a Multiomics platform over the years. It features Lipidomics. Today, it also allows us to do flux analysis. I will not have time to go into that. Since we have a mass spectrometer, we thought, why don't we use it also for doing proteomics? And we could also do phosphoproteomics. It's basically what we've been doing so far. We have other studies where we've been doing RNA-seq. Don't have time to go into that. But what is overall arcing our platform is actually that we set it up such that we are able to typically do time series analysis of physiological processes, meaning that you need at least 100 samples to look at dynamic changes over time. And when you want to do that, you also need to have a system installed that allows you to do computational analysis of all of this high content data, because otherwise you don't get anywhere. And to make it quite clear is you don't want to do this in Excel. So you need other kind of tools to basically get headway with this. So the platform that we have established and the way that it looks today is summarized here. As I told you, that it has a lipidomics element, it has a proteomics element, and it also has a phosphoproteomics element. We've been validating it for yeast, mice, and we also been lately been doing some analysis of human samples. You take those samples and then you either go into a lipid extraction to do the lipidomics, or you do a protein preparation or protein digestion to start doing basically proteomics, or you can do an IMAC, offline IMAC enrichment to isolate the phosphopeptides, and then you can do phosphoproteomics. Yeah. So on each stage, we basically have over here in the lipidomics aspect, and I'll get back to that in the next few slides, is that we're basically doing high resolution mass spectrometry with an Orbitrap fusion. We push the data into our homemade software called ALEX123, and then we do lipid identification and quantification. On the proteomics side, we basically use the same mass spectrometer with an LC system in front. And then we basically do PD and progenesis, identification and quantification of peptides, proteins, and phosphoproteins. And then at the end of the day, you sit with a lot of data, and then you need to do some data integration and also data visualization to basically, interpret your data. So I believe that the lipidomics is more novel to you. So I'll give you a few insights into how we actually carry out that work. Before I do that, I'll just give you a quick dictionary of how you actually talk about lipids. And to talk about lipids, we typically apply a so-called shorthand notation. So what you can do in your chemistry program is that you can draw a molecule. In this case, I've drawn a PC, phosphatidylcholine molecule, abbreviated PC, with a 16-0 chain on the SN1 position and the so-called arachidonic acid chain on the SN2 position. And this arachidonic acid has 20 carbon atoms and 4 double bonds. And if you count from this carboxyl terminal, you will see that there's a double bond at the position 5, 8, 11, and 14. And they're all cis double bonds. Now here comes the issue is that in mass spectrometry, we typically measure masses. And since we do high resolution measurements, we typically get it with 4 decimals, measuring the monoisotopic masses. From that information, the only thing we in theory we can do is basically to compute the so-called molecular formula, basically how many carbon atoms, hydrogens, oxygens, nitrogen, and phosphorus does it have. And now with the help of a database, this is the same you would do in proteomics, you can then map that to some structural feature of the molecules you're interested in. In this case, it's a lipid molecule. And we know in the database that this PC molecule that has a total of 36 carbon atoms and 4 double bonds would have exactly this molecular composition. Yeah? But just measuring the intact mass doesn't give us any information about what are the fatty acyl chains, not at all. So to do that, you actually need to fragment the molecule. And when you do that, you can then annotate the molecule at a different level because you now get the information. In this case, it would be 16-0, 24. But just with an MS2, fragmentation, you don't know where on the SN1 or SN2 the fatty acyl chains are sitting. It could be the isomer, where the 24 is sitting on the SN1 position and with 16-0 on the SN2 position. So to denote that, we basically use a dash. And also notice that I don't indicate the double bonds because just measuring the MS2 information doesn't give us that information. For that, we need to do other kind of, analytical procedures to get that information. And this is what I call the full molecular composition, which today is not really possible with a single acquisition method. So that's something to be aware of in the analytics or lipidomics. Okay. The instrument that we have is an orbitrap fusion. It has a lot of nice features, high mass resolution going up to 500,000 in the orbitrap mass analysis. I predict that most of you are aware of this. It has a quadrupole mass filter that allows us to do very nice isolation of basically our precursor lipid ions to do fragmentation. It has 2 fragmentation cells, the HED fragmentation cell and the CID over here in the area of the ion trap. And then we have another detector, which is the so-called ion trap detector, which can be operated at very high speed. So this is what you can call a tribrid instrument, 3 mass analyzers and 2 detectors in one box. So that allows us to do a lot of things. In front of that, we have a so-called transverse nanomate sitting that is pushing in the ions. This is basically how it looks. It has you put in a 96-well plate, then it has a rack of conducting pipette tips. You program it to go to the well A1, suck up 5 microliters of sample, and then go to the orifice of the mass spectrometers where there's a small little chip sitting. And this chip has an array of 20 times 20 small little nozzles through which you spray your sample. So you don't use a new needle for every, you use a new needle for every injection, meaning that there's no sample carryover when you do this, compared to what if you would do, for example, flow injection. Here's a depiction of how it looks. This is the ESI chip. You have these nozzles through which you do the electrospray ionization. In the tip, you have your lipid extract, typically in chloroform methanol, and you basically put the tip onto the chip. You put a bigger bed pressure, you turn on the voltage, and then basically you get ionization either in positive or negative ion mode. Yeah. So what is this high resolution actually good for? So here I made a mixture of 2 triglyceride molecules. Some of you might know that triglyceride molecules are what make you obese. They're also what you are walking on. This is a fat pad under your feet. I have one molecule called 54-2, which has the monoisotopic mass of 904. And then I have one molecule that has 1 double bond less, 54-1, which has the mass 906. And these two molecules are mixed 5-to-1. So they're synthetically available. So it's a synthetic experiment. Now, if you go back to 2005 and 6, basically the time when I was in Dresden with Magno, we had access to a so-called LGQ Orbitrap XL mass spectrometer, which was new at the day and was very exciting. If you inject this mixture on that kind of instrument, you will see in red basically the measurement, and in black you will see a simulation. So for the first molecule, the M0 of 54.2, you'll see a nice Gaussian distributed peak. If you now go to the first isotope, remember that you always have carbon-13 isotopes in nature. You will also see in red a nice measurement and also a nice simulation. But now when you go to the channel 906, we start having a problem because what we measure is actually underestimated. And from a fact, we know that there should be 2 molecules, but we only see 1 peak. And we actually see that the resolution of this peak is slightly broadened. And sorry that I forgot to say that the mass resolution here in the Orbitrap XL is basically 86,000. Now, when we go to the Orbitrap Fusion, came out around 2014, 2015, we inject the same mixture, and you now get an effective mass resolution of 235,000. Now see that the peak gets much more narrow. Red is measured, black is simulation. First monoisotope is great, first isotope is also great, but now when you go to 906, where we know we have 2 molecules sitting, we basically see in red what we measure, that we almost get baseline separation of these molecules compared to what we would expect from the simulation. So on one side, we basically can baseline separate individual molecules with high resolution. It also gives us an avenue for doing flux analysis with stable isotopes, which you cannot really do with lower resolution instruments, or might be quite tricky. So I'll just get to one point is that why would you want to waste time on doing LCMS? If your mass resolution and fragmentation of your mass spectrometer can separate distinct molecules. So this is typically what you would do. This is typically what you see in your textbook on mass spectrometry. LCMS, you have an LC system, ion source, and mass spectrometer. In time, you separate molecules, they come off your column, and then you basically take the molecules and everything is good. Yeah? But why would you do that if you could now just eliminate the chromatography system, just inject your sample and baseline separate the molecules with a high resolution? And this is basically the core elements of shotgun lipidomics, is to remove that LC system. Don't waste time on it, but use the mass spec solving power and the fragmentation capabilities of the mass spectrometer to get to identification and quantification of individual molecules. So you might say, well, LCMS is of course much more quantitative and it's so much better. So here's one example where we basically testify how good and accurate is high resolution shotgun lipidomics. So here's another triglyceride molecule called 51.3, which is titrated over a constant amount of the internal standard 51.1 with a D5. And we basically record FTMS data. So here you see the data points in blue with a 2-fold dilution series with the molar ratio of the 2 analytes. And you see here a line which has the slope of 1. And you basically see that you have a 1-to-1 relationship between what you titrate, your analyte, and then the internal standards. You have a limit of quantification in the range of 1 nanomolar on chip, and you have a dynamic range in the order of 10 to the 4. Yeah. So in other words, the instrumentation is highly accurate and it's also highly precise. And I don't have any data to, well, I have data to show it, but I left that out. So how do we reuse the machine? Again, we have eliminated the chromatography system. So what we do is that we have a method that we call MS-all. It involves recording a high resolution MS1 scan. After that, you basically record MS2 spectra with a 1 Dalton step, typically in a mass range from 400 to 1000. So you basically shoot on everything. So machine gunning everything in 1 Dalton steps. Yeah? So here would be the example. You get to 500, you get to 501, you get to 502. You just keep shooting on everything. And it doesn't matter because you have time And you also have, how to say, the possibility of doing it in the shotgun approach. Now, when you get to 731, you see in the MS1 scan here, there's a sphingomyelin molecule with 36 carbon atoms, 1 double bond and 2 hydroxyl groups. You fragment it. Now you see a 184 fragment that tells you it's a sphingomyelin. And if you zoom in around 260 and 280, you will see 2 structure-specific fragment ions that tells you what are the 2 chains of this sphingomyelin molecule. So it's an 18-1-2 and an 18-0. You can do the same if you get to 760, the most abundant PC molecule in mouse brain. You will see now that you get a 184 signal also. It tells you it's a PC molecule. And when you zoom in around the mass range from 400 to 600, you can see there's an 18-1 chain. And there's also a 16-0 chain that allows you then to say 16-0, 18-1. And you continue like this. I'll just give you one example here again, 810, PC, 38-4. And when you now look at the structure-specific fragment ions, you get 2 isomers, 18-0, 24, and 18-1, 23. Yeah. So of course, you don't want to do this manually. So for that, we've been developing software over the years. It's called Alex123. And basically, it works in the following way that you have raw data that you acquire on your thermo instrument. You push it into the so-called Alex123. This data you push forward in something called SAS, and then you visualize it at the end of the day in Tableau. So we have a database underlying this with more than 430,000 different lipid molecules, including fragmentation information. You need to tell our software what you want to look for. This is one of the caveats of our system, is that you need to have some biochemical intelligence to use the software. If you don't know what you're looking for, you can also cheat the software, but this is a little bit more for advanced users. You then tell the software to go and identify the lipids you're interested in. Then you basically use the SAS to do quality controls, quantification. At the end of the day, you then push the data to this software called Tableau for doing visualization. And what is good about Tableau is it's dynamic data visualization. Yeah. Okay. So that was basically a wrap up of the lipidomics system that we have on our platform. Just give you one, how to say, outlook of what is coming and what we're working on, is that we're basically now piercing a small little hole in our mass spectrometer. And then with the help of Stephen Blanksby and the colleagues in Brisbane, Australia, we are pushing ozone into the ion trap element of our mass spectrometer. Yeah? So we have an ozone generator, we pierce a hole, and now we deliver ozone into the ion trap. So what can you do with that? You can basically get double bond precision of your molecules. So here's an example. If you have a molecule, PC 16-0, 18-1, commercially available, you get told from this provider that the double bond is sitting on the 9 position, and you also get told that it's a cis double bond. Now, if you do an MS2 scan of this molecule, you will see the fragment ion 281.24. Just this information doesn't tell you where the double bond is sitting. It doesn't tell you what orientation the double bond is. It just tells you that you have a fatty acid chain with 18 carbon atoms and 1 double bond. It doesn't tell you more. This is one of the key things you need to know in lipidomics. What information do you actually can you extract from fragmentation information? Now, if we now do an ozone reaction of this 281 molecule or fragment in the gas phase, and then we record an MS3 spectrum, you will see that the ozone is specifically reacting on the position 9 double bond. And we'll get 2 fragment ions, one at 171 and another one at 187, which you see detected here. Now, if you move the double bond down to position 11, you will get a different mass, which would correspond to you having the double bond in a different position. So with this kind of new technology, we actually will be in the future able to get double bond precision on the different fatty acid chains. But so far, it's still not really available as a standard practice. So time is running. So in the last part of the talk, I'll just run you through some work that we were doing. It has been many years under the way, and the manuscript is currently under revision. Basically goes into studying circadian regulation of lipid oscillations in liver and in plasma, and trying to tease out what are the molecular relationships between the liver and the plasma? Can we use the plasma to get insights into what is the liver doing? Or in other words, what lipids in the plasma tells us about what happens in the liver? And that's of course relevant if you go to the clinics where you will do a plasma measurement, and then from that could you actually tease out whether the patient has a problem with the liver. So what we're particularly interested in is basically diurnal rhythms. And you might be aware that you just got up, most of you in Brazil, you maybe had a coffee with a lot of sugar inside, and you might remember that that basically kicks in insulin signaling. Insulin will get released from the pancreas, and that will drive a lot of processes in the liver and in the fat tissue and also in the muscle tissue. One of the things in the fat tissue is lipogenesis. In the liver, you see glycogen synthesis, you will see cholesterol biosynthesis. You might also ask yourself the question is that, well, if there's a lot of sugar coming into my blood veins and it comes to the liver, should I not also turn on lipogenesis in the liver, same as in the fat tissue? Yeah. Conversely, if you go to sleep in a few hours, I will be there in probably in about 4 or 5 hours. Then we stop eating, insulin level goes down, glucagon gets released from the pancreas, and that triggers another set of reactions, or how to say, processes in our different tissues. Basically, in the fat, you will start seeing increased level of lipolysis and release of fatty acids from the fatty tissue into the bloodstream. In the liver, you will see an increase in the so-called gluconeogenesis, glycogenolysis, and also mitochondrial biogenesis. And the fatty acids that comes from the fat are going to be absorbed, converted into triglyceride molecules, and then packaged VLDL and sent out to the circulation again. So these are some of the things we know. So what we were basically interested in, can we somehow use our Multiomics platform to study circadian regulation of lipid metabolism? To that, we were not allowed to kill any human beings, so we went to mice. What you need to know about mice is that they are nocturnal. So basically, they are opposite to us. So they are awake and feeding during the night. And when there's light in the stables, then they typically go to sleep and they are relaxing. Yeah. So just keep that in mind when you see the data. So what we did to do for this study is that we collected different tissues. I will show data from liver and plasma today. We did it over 24 hours in such a way that we had 8 time points across the 24 hours. And in the first day, we collected tissues from 3 mice, and then we repeated it on the next day. Yeah. So like that, you either have 8 time points with 6 biological replicates over so-called 1 circadian cycle, or you have 16 time points with 3 biological replicates across 2 diurnal cycles. And for this, we used a total of 48 mice. So all this, we got basically pushed into our Multiomics platform, acquiring all the data and putting it together. And of course, over the years, then we were doing a lot of data interpretation, correlating the data and trying to see what comes out of this. So I'll go a bit quickly through it. I don't know how much time I have left. Just mention that we compare ourselves to a study in 2014 by Adamovich et al. What do we have in terms of lipidome coverage? They basically had so-called 4 categories. We have 5 categories. They quantified 159 lipid molecules. We have 1,030. You can do some statistics. It has to be done a little bit differently. We can get back to that later, but they found 27 lipids oscillating. We find 562. If you look at what changes, you'll see a lot of the changes are in the so-called glycolipids. That means your triglycerides. There's also things happening in your phospholipids. And what was particularly interesting, and we'll get back to that, is that we saw diurnal isolation in very low abundant MMPE and DMPC molecules, yeah, which are basically a intermediate product in the PC biogenesis in the liver. So here's another example of our data, comparing it to the 2014 paper. This was done across 1 diurnal cycle with 4 biological replicates. This was the most circadian lipid that they reported. It goes up during when they are, how to say, relaxing during the day, when the mice is primarily sleeping, and then it drops down. And then when it becomes dark, the mice get active and they start feeding again, and then it comes up. And they told you that this could be one of two PC molecules. When we do the analysis, we find 3 individual molecules, 16.0.23, 18.1, 18.2, 18.0.23. And we can display that data either on a 24-hour axis or on a 48-hour axis. And like that, when you do it on a 48-hour axis and calculate the so-called Pearson interday correlation, you can see that it is truly oscillating, meaning that this is repetitive and it's not just random noise that you have. Then you can go and use cool, mathematical procedures like VS-Clust from Veit Schemle, and I know that you heard a talk from him a few weeks ago. So this is what we do. When we use this algorithm, we basically find 4 clusters that we call LL1, LL2, LL3, and LL4 for liver lipid. You will see that there are molecules that go up while the mice are fasting. Then when they start eating, it plummets down. This other group is going more gradually up during the fasting. It stays for 2 hours in the liver, and then it goes down. The LL3 is down during fasting, and then it shoots up immediately upon food intake. And then we have the LL4, which is more gradually decaying over the fasting period. And then it stalls a bit when the mice eat, and then after 2 hours, it starts shooting up. So kind of different dynamics we have. If you do hierarchical clustering, you will see that these different time points that we have are separating into 3 different, periods. We have fasting, 4, 8, and 12. Then we have data for 13, 14, which is correlated to when the mice are feeding. And then we have these data points, 16, 20, and 24, which is now what we correlate to the so-called postprandial state. So with that, you can get information about what is actually, interesting about these different clusters. You can see that LL1 is what we can call early fasting related. LL2 is late fasting related. LL3 is directly feeding related, and this LL4 is much more postprandial related. But you might say, well, very good, but what do you learn from that? And just looking at these different molecules didn't tell you so much. So basically what I developed was another way of doing enrichment analysis. Most of you know that from the Go analysis that you will be doing with your proteins. I have to say that I was very much inspired by a paper from Martin Mullenar that was published in GigaScience in 2019. Some things were slightly, not working with the data that we had. So I had to basically re-engineer it for our own purpose. And I did that in SAS. And basically what we do is that we have lipids in these different clusters. We have all the lipids that we have detected. And now you can determine different frequencies of so-called structural attributes. Yeah? It can be lipid category, lipid class, fatty acid chain, or what we call the backbone, so-called some composition. And this frequency you can either calculate for the cluster or for the overall occurrence in your data set. And now you can do a simple Fisher's exact test. And now you can test whether the frequency of a given feature in your cluster is more overrepresented or underrepresented compared to what you have in the list of all the things that you have detected. So here's a case of the LL4 cluster that has a lot of PI molecules. So the frequency is 30%. When you just look at all of the molecules we detect, is around 5% that are PI molecules. With the Fisher exact test, we can basically see that we have a significant overrepresentation of this group of molecules in our cluster. And then we have this odds ratio of basically saying that it's 8.1 fold more likely to find a PI molecule than by random chance. Yeah. So what we can do is we can visualize this, basically go a little bit fast through this, but just push your attention to the so-called early fasting related LL1 lipids. They enrich in triglycerides. They're basically enriched in 18.2 lipid, and then these other multiple double bond containing backbone elements, 47, 48, 42.9, which are basically structures that are related to a 22.6 fatty acid. Conversely, this late fasting LL2 cluster has a higher, is enriched for 24, which is arachidonic acid. The feeding related is also enriched in triglycerides, but you will see that this is basically enriched in saturated and monounsaturated fats. And what is also interesting is this LL4, postprandial related, is basically depleted in triglycerides and heavily increased in phospholipids, PI molecules, phosphatidic acid, and also this DMPE molecule that I will get back to. So this is basically what I told you. What are the enriched features? So this basically raises a few questions of where does these 22,6 fatty acid chain comes from in the LL1 cluster? Could it be here in the LL3 cluster? Since we have saturated triglycerides, could it be that the liver is doing lipogenesis from the sugar that it gets? And how can it be that we have a cluster of only membrane lipids that are being somehow pushed out of the liver? Yeah? To look at the connection to the plasma, basically what we did lipidomics of plasma. We found a lot of lipid molecules. I'll just briefly go through that. Just highlight that we also found for the first time these unique DMPE molecules in the blood plasma that has never been detected before. Yeah? And again, these DMPE molecules are intermediates in the synthesis of the PC molecules via the methylation pathway in the liver. So this will become very important as we go forward. So here you have basically the clustering we did with Veit's routine. We also found 4 clusters for the plasma, PL1, PL2, PL3, and PL4 for plasma lipids. You can do hierarchical clustering and you will see that you see the same periods, fasting, feeding, and postprandial state in the plasma. You can do the enrichment analysis and you'll see that the PL1 is enriched in molecules with 22,6 fatty acid chains. Yeah. You will see that the PL2 is enriched in cholesterol esters. This is typically what you have in the LDL pool, but these molecules have now 24 fatty acid chains. You will see here in the PL3 that you have in the plasma triglycerides with 16,0 and 18,1. And what you will see now here in the plasma is that in the PL4, we have triglycerides with 18,2 and 18,3. So let's just quickly benchmark that to what do we have in the liver. If we stay here in the PL4 and the LL4, you see in the liver that you have mainly triglycerides coming up, but in the liver, we have membrane lipids coming up, okay? It's a little bit counterintuitive because if something comes up in the plasma, why doesn't it go up in the liver? And I will go back to that in a little while. However, here in the PL1, what you basically see is that the liver or the plasma has an increase in 16,0, 18,1, and you also see that in the liver. Okay, that's pretty interesting. And again, PL1, if you go over here, we saw in the liver that we had an increase in these 22,6 containing molecules in the liver. And now we find that in the plasma, we get an increase in molecules with 22,6. So in this case, could it be, and this is what we want to test now, is could we test if these predictions that the 22,6 in the plasma is what goes into the liver, and conversely, what is the axis that we see here in the PL3 cluster? Yeah? To do that, I personally developed a routine that looked for so-called crosstalk between the different tissues. And that crosstalk is basically defined as a temporal correlation that is better than 0.66 in terms of a Pearson correlation score between identical lipid molecules or lipids with identical backbones in the liver and the plasma. So here's a depiction of that. In the LL4, we have in the liver DMPE 16,0, 18,2. In the blood, we see the same molecule with a Pearson correlation of 0.98. This is pretty damn amazingly good correlation, I tell you. Yeah? At the same time, we also see a PC molecule 16,0, 18,2 with a correlation of 0.84, which is also a very good correlation. So this is what we define as crosstalk. Yeah? Here's another example of the LL1. We have a molecule 16,0, 25, 22,6. We see the same molecule in the blood plasma with a correlation score of 0.88. We also see this non-esteroid fatty acid 22,6 having a correlation of 0.73, suggesting that this fatty acid could come into the liver, get assembled, and then kicked out again. Yeah. When you now look at this crosstalk based on the different clusters, LL1 to LL4, you would see that we have around 50 molecules in the LL1, not so many in the LL2, 50-ish also in the LL3, and basically 64 in the LL4. Now, when you look at how does this actually look when you, I would say, compare these identical or similar molecules, and to do that, I'm doing a so-called Sankey diagram where we have the common lipid class features. Remember that LL1 is enriched in the triglyceride molecules heavily, And then we're looking at what are the fatty acyl chains that are elevated here. And you see that 22,6 is one of them. And now if you correlate the temporal dynamics, you will see very nicely that this set of 22,6 containing molecules comes up in the plasma early. And then there's a 2 to 4 hour offset until it reaches the liver in the LL1, and then it drops down. So that basically tells us that there is a release of 22,6, presumably from the white adipose tissue, and this is taken up by the liver, as you see in the LL1. It is then converted into triglycerides, and it's being pushed out as a VLDL particle. I'll skip the LL2. If you look at the LL3, here I told you that these are also triglycerides. In this case, they're highly saturated, highly enriched in saturated fats and monounsaturated fats, is what you can see, 18.1, 16.0. When you look at how does that compare between the plasma pool and the liver pool in LL3, we basically see in the liver, it shoots up right immediately when they get food. And then there's a slight delay in the increase of these triglyceride molecules in the plasma of 1 to 2 hours, suggesting that these molecules with saturated fats are being made in the liver and are then being released into the bloodstream as VLDL particles. That's basically what I summarize here in the text. Now, what about the LL4? What have we learned? When you look at that, these are only coupled phospholipid molecules. When we look at what happens after they are eaten, here you see it in the blue trace in the liver, there's a 1 to 2 hour delay, and then they shoot up, coming out into the plasma, and you basically see a 1-to-1 correlation in the temporal dynamics, suggesting that these membrane lipids are being pushed out from the liver directly into the plasma. And what we know from the textbooks is that this is actually a feature of so-called nascent HDL production from the liver. These are one of our other lipoprotein particles. And what I basically want to draw your attention to is that these, what we have in these cluster LL4 is the DMPE molecules. So these are the intermediates in the PE methylation pathway. So how do we make some biological sense of this data? So one way to do it is to look at the PE methylation pathway. So here is drawn the enzymatic cascade. You have interleuramine, phosphoetanolamine. These are basically enzymes involved in the pathway. Then you couple that to a so-called, you incorporate that so you get a PE molecule, and now you carry out 3 methylation reactions with the so-called PMT enzyme, and then you get PC. And then what is known from the literature is that that is somehow coupled to VLDL biogenesis and secretion, because if you knock out the enzyme, if you knock out PMT, you have a problem making ApoB or VLDL particles. Also note here that we have other enzymes that are replenishing the pool of these S-adenosylmethionine. Yeah. If you look at basically the MMPE, the DMPE and the PC levels, both in liver and plasma, liver in blue, plasma in red, what you can nicely see here for 16.022 is that it drops down during fasting. Once the mice eat, it shoots up. And you see the same happening here in the DMPE in the blue. This is what you need to notice. Now, what was striking that when we found this is that you actually now, when you look at the trace of the plasma, is that you more or less see a 1-to-1 relationship between the liver and the plasma, suggesting that this DMPE molecule is being released from the liver. Also, when you look at the downstream product of the DMPE, which is the PC molecule, you will also see that it has a similar distribution or profile in the liver, or sorry, in the plasma, but not in the liver. Conversely, if you go to this other molecule, the so-called PC 16.022.6, you see something completely different in the plasma. Yeah? You see that this PC molecule comes up during fasting and then it drops down immediately after eating. But now when you look at the precursors, the MMPE, it basically goes down during feeding, it goes up upon eating. If you look at the DMPE, it has the same, temporal pattern. But now if you look at what happens in the plasma, you basically have an inverse correlation, that it comes up during fasting and then it drops down, exactly like the product PC. But the overall level stays of the PC molecule stays constant. So it's a little bit intricate, I must agree, but basically what it tells you that is you have 2 different pools of PC molecules with distinct diurnal acylations in the plasma, which is shown here down in the red, suggesting that this is basically mediated by the PMT pathway. So to examine that, we went into the proteomics data, and I also have some RNA-seq data where you basically see that the PMT expression goes down during fasting, comes up after eating, and you basically see the same for these other accessory proteins that you have in the PE methylation pathway. And you also have this ApoB, which is also related to the PMT pathway. So we can basically conclude that the PMT methylation pathway is diurnally regulated, not only at the single enzyme level, but also on these auxiliary enzyme systems. And that prompted the hypothesis whether these oscillations we see in the DMPE and the PC molecules in the plasma that are highlighted over here, whether they are truly due to PMT activity. And to basically examine that, we went into the knockout mouse, PMT knockout. And I will not share the data with you, but basically take-home message is, yes, we have an effect of knocking out this enzyme. So we can conclude that this pathway is diurnally regulated. So coming to a summary, I hope I have convinced you that we have a pretty decent multiomics platform and that we like to use it for doing systems biology studies of lipid metabolism. It includes proteomics. We also do phosphoproteomics. I showed you basically some of the key tenets of the shotgun lipidomics platform that allows us to do comprehensive and quantitative lipid analysis. It's accurate, it's precise. You can also take the same software infrastructure and push it on to do lipid imaging by molding if you want. Currently, we are also, with the data that we have been then developing new bioinformatics tool. And then what we're trying to do in the future is basically to extend the technology to do double bond precision with ozone reactions in the gas phase, trying to do flux analysis also with instrumentation and the high resolution. We have a few high quality time resolved multiomics resources available if you want to do, more discovery work and trying to study regulation in terms of lipid remodeling in yeast, phosphoregulation also in yeast, circadian regulation, the data I just showed you. And then at the end of the day, one of the examples of discoveries is that we have this hepatic PE methylation pathway is diurnally regulated. And it really opens the door into doing time series analysis of human blood plasma. And maybe we could find some biomarkers that are basically predictive of metabolic health. So I thank you very much, Brazil, for your kind, for the possibility to share the data with you and also for your attention. And I'm very happy to take any questions. So that's it.
Thank you very much, Christian. It was really, really, really nice. I will tell you that you have a damn good omics platform. That's really, really comprehensive. I don't know if I can start with the questions or Magno is, if you want to start. We already have some questions from the audience. Okay, so Magno didn't say anything, so I'll start. I have 4 questions, more general, that I'm going to sum up for you, if you don't mind. The first 2, it's more related to the technology itself. So first, if you would say that you're already happy with the resolution that the vendors are giving to you to increase resolution, it will be like needed for the way that you are this LC-free method that you develop, or you'd say that 200,000 is fine, you don't need more than that. Regarding, I was really interested about this ozone fragmentation technique. I never.
But should we take the first question? So that's a really good question, and also it's a very common question in a sense. So there's different things related to this question. That one thing is that it always pays off to know exactly what you want to achieve with the studies, and sometimes you don't need high resolution. And flux analysis was already done, 20 or maybe 18 years ago. It was already done on low resolution, triple quadrupole instrumentations. So if you have a question that relates to those kind of studies, you can still use it under those settings. But some of the things that we're doing today, especially with the fatty acids that we're labeling and giving them to cells, it's a bit difficult to do it as the way you did it in the old days. So for that, you need more specific techniques, and there you need the high resolution. So the question is, how high do you need the resolution? And to make a long, complicated answer, kind of simple, is that it kind of depends on how many, how to say, stable isotopes you put into your molecule. Yeah. So depends on how to say, if you're doing deuterium or you're doing carbon-13 labeling, because they will have slight differences in the mass offsets. So you can, for example, use 5 deuteriums and you can get by with a 200,000 resolution. But if you're doing 5 carbon-13 atoms, you should be a little bit careful with doing that. So these are some of the technical things you need to be aware of. And then maybe another element of that is, do we need to go higher? One element of that is that it's always nice, but I think we are somehow more or less saturated with the 500,000. 200,000 is still pretty good for many things. And again, it depends on what you want to study. And then I try to fully answer the question is that if you cannot resolve the molecules by shotgun, of course you need an LC system. So that's also where you need to know what do you want to achieve. So if you want to achieve very low abundant molecules that are suppressed by all the other molecules, you need to go to an LC system. But if you want to do what we are doing, studying bulk amounts of lipid and how cells are regulating bulk amounts of material, you don't need super sensitivity for something very low abundant. And it also basically, we can also do pretty cool stuff already with tracer isotopes. So we are still pretty, how to say, sensitive. So I think that hopefully took care of most of your questions.
Yeah, great. I will skip for now the ozone question, and then depending on how it goes with the audience, I come back to that. So more like for the biological meaning, I don't know if you're already studying that or you plan to do that related to how the microbiome works in the lipids metabolism, if you're considering to study that, and also related to, because it was really cool, this oscillation that you've shown, and I wonder if you are planning to kind of challenge your animal model to shift, like trying to mimic this night shift workers that we know that these guys have a lot of trouble with metabolism, and also sometimes they can develop like diabetes and a lot of metabolic disorders. And if you are planning to study that.
Yeah, that's definitely on the drawing board. I would say maybe it wasn't 9 years ago. It was a few years ago when we started doing this, and before we opened up the data, you didn't really know what it would mean. So you somehow, this is what I think was pretty, I would say, amazing and also motivating, is that when you unfold this complicated data, you start learning new things as a mass spectrometrist. As Magno pointed out, I was a little bit more of an analytical chemist, but having data sets like this, you need to read up and do your homework on circadian rhythm, fasting feeding, insulin signaling, and all these things. And of course, out of that, you see why you would want to go and do more complicated work in mouse models. Looking at, for example, the feeding time is one of the things. It could also be high fat, how they obese mice, or diabetic mice, or even humans. Yeah, there are many possibilities now. So that's for sure on the drawing board, and anybody who would be interested in working on that could, of course, contact me. So
great.
Yeah.
We do have a question for, I think it's a friend of you, Petrus from the Lipidomics. I don't know if you can see the question.
Yeah, so in the comments there are, I don't know what.
It's also here in the screen, in the monitor. But yeah, he's asking about this odd chain fatty acids, if they come from the diet, and any idea why not pick it up as an excellent PowerPoint.
Well, we do, and I have to confess a bit, in the sense that the data that I showed you, has been have been underdeveloped for a bit of time. And back in the day when we did the searches, we'd only search for even chain lipids, which are the most abundantly in nature. Yeah? But since then, you will also see that it's already published with my colleague called Robin Clem in Molecular Cell a few months back. where we're using exactly the same platform, but now we basically open the search space for doing odd chain, triglycerides, everything odd chain, and we don't see any problems in the, how to say, in the identification and the quantification. So we are able to take that apart computationally. And there's also another paper in the preparation that's coming out where we do CRISPR-Cas9 knockouts of cells. And basically we can nicely see when cells differentiate how these odd chains molecules comes up in adipocytes. So we make them ourselves. It's not only from the outside, but it's also endogenous metabolism. And this is like a time series experimentation where you clearly see that, which I think very nicely shows that our technique is specific for that. But again, I didn't have time to reprocess all the data. I hope the reviewer is not necessarily listening to what we're saying. But it's quite a bit of a, how to say, undertaking to reprocess all the data. But
yeah,
it can be made available if people want to do the processing. Let's put it like that.
Lais is asking you, like, she's like, of course, like congratulating you and asking if this Lina is an open source tool. This software, is it Lina? Yeah.
Yeah, Lina. So that's also the name of my daughter. That's why I couldn't resist myself
Yeah.
to pick that acronym. So it's basically, it's a script that I'm running in SAS. And if people are interested in it, I'm more than happy to share it. Okay. But it requires that you're able to run SAS. That's basically my scripting language.
Okay.
That's what it is. But I would say that more or less the tool by Martin Molinar, the Lion, does something very similar and should be good enough for most purposes. So. Okay,
so maybe Lais can contact us and then we give her your email. You can also search in the, yeah, she can also search in the Google, right? Or if you want, you can write it. I don't know if you want to share it public here in the YouTube. I don't know how you prefer to do that.
Well, let's get the paper through.
Yes.
And then it's but let's just contact me if somebody is interested in that script, then I'm happy to share it.
So Lais and who else wanna use this tool? Contact. Great. So Giuseppe also has a question for you. Yeah. So Giuseppe is now. Let's say that was the question that I saved. So thanks, Giuseppe. So you want to see the implications of this ozone fragmentation? Yeah, I confess that when you say that you punched a hole in the instrument, I also cried quite a lot.
Well, we're basically, it's the helium gas as far as I know, where we basically have a splitter and then we can purge it in with ozone, and then you can also regulate the level of the ozone. But here you basically see this is an FTMS3 data set, yeah, and there's no zoom on the MZ axis as I show now. Let me just put the pointer on.
Again. Wait.
So I would say maybe it's 50, maybe 60% efficient if you sum up these intensities. So it's pretty good, and yeah, let's leave it at that.
Does it also change the pattern of the fragments that you form, or it's just like efficiency, or you generate other type of ions? I didn't get this point.
With the ozone, or
yeah, with the ozone.
It's still in its early stage, and one of the challenges that we have is that when we have a lot of double bonds on the molecule, then we have seen that things can start scrambling. Yeah.
Okay.
But we don't know that, because also when vendors are making standards, it's very difficult to control their purity, because most of the purity work is done by a GCMS analysis with a conversion to a fatty acid methyl ester, which involves typically a high temperature trans-esterification reaction, where you might also have rearrangements of the double bonds. And even if you have 5% contamination in your organic synthesis, would you be able to pick that up on a GCMS trace, which is only maybe 15 minutes long? And most people, as far as I can see, and I have to be careful what I say, but I would be very happy if the companies would do 120 minutes GCMS analysis on their synthetic standards to allow us really to see the purity of polyunsaturated lipids. And with that information, we would be more sure whether we have, how to say, gas phase, how to say, rearrangement with ozone, or whether it's just basically a purity issue from the vendors. That's still an open question.
Okay, and scary answer, I would say. Okay, Lais also, she has another question for you, asking how do you integrate, and it seems to be very labor, how you integrate the proteomics, phosphoproteomics, and lipidomics data?
To the take-home message is we basically did it all in the brain.
Okay, yeah.
So I say I there, as far as I know, let's say we try to do correlations. I try to do correlations in many things, and I didn't really find it that informative. And it goes a little bit back to the central dogma slide I showed in the beginning, that I think for some phenotypes, some functions, it's very difficult, even with a proteome lipidome correlation, because in some cases it will be enzyme activity, and maybe if you're lucky, that enzyme activity is phosphoregulated. So only if you do phosphoproteomics, you will be able to pick it up. But if you don't do phosphoproteomics, you don't know. And also, as I showed in the data for the RNA-seq, for the circadian rhythm, is you can't see anything out of the RNA-seq in terms of what we found. So that means that the enzyme activities are somehow not really recorded in the RNA-seq, and they're also not recorded in the proteomics data that well for most cases. Just to highlight what I said, just zoom ahead. I don't know if you want to share my screen again. Here it is. So if you look at like the ApoB, so we see the proteomics kind of constant, and then there's a drop down upon eating, and then it comes up. If you look at the RNA-seq data in green, it's a flat line.
Yeah.
If you do the RNA-seq on PMT expression, it jumps a little bit, but it doesn't really get you to the conclusion. So in this case, of course, it's good to have proteomics. It guides you, it's useful. And then you also have the other proteins in the pathway that helps you. But very often you will also see scenarios where you're sitting with RNA-seq or proteomics data, and you can't explain why something oscillates on the lipid level. So in other words, it's not that easy. And take-home message is that we did try to do a lot of correlation work, didn't find it so informative. So there was a lot of, how to say, biochemical intelligence, and basically going in with your biochemistry goggles and zooming out on the things and cross-correlating the things. And for that, we use Tableau, which allows you to do things very dynamically, clicking on buttons. Compared to doing Excel, I think you would probably not want to do things like this in Excel. But where there's a way, there's a will. So everything is possible.
Yeah. There is a question from Genilda from our WhatsApp group. She's, if I understood correctly, I think it's something that you're going to study now, because she's asking if there is some disease signatures that has more or less the same trend that you're showing. So as far as understood, that the next step is to search for kind of markers, right, that could be used to diagnostic markers that oscillates, right? If I understood correctly.
Yeah, but probably the clinics would argue that this is not relevant for humans. So in that sense, it's nice to do it in animals. Mm-hmm. So my animal work would be more going for more, how to say, basic science and studying, how to say, effects of diet or age. We also have certain tools where we can specifically knock down the expression of certain proteins in the liver that was done in a collaboration with Roche in Copenhagen. And I rather want to use those tools for very basic science investigation of what happens if you knock down certain enzymes in the liver. And the question is whether that translates to medicine. I don't know. That's a little bit early.
Yeah. Just a curiosity of mine. Considering all the time, of course, it depends from people to people that are like taking their study. But I would say more or less on average, how long it takes this kind of study if you want to integrate lipidomics, proteomics, phosphoproteomics? Are we talking about like 1 year project, 6 months project? How long it takes?
Very good question. I need to go back and check when we acquired the data files. That would be, and then calculate. I think for us it took a few years.
Okay.
And I think if you have a single person sitting on it, or let's say you need probably 2 persons sitting on it, I think you can probably do it in 2-3 years.
Okay.
Acquiring the data, but if you also want to do the mouse work, right?
Yeah.
And now we've been through it once, so we somehow know what not to do. We would be faster. And we also there's a lot of things we didn't have a lot of data processing pipeline elements for the lipidomics we didn't have available back in the day. And this was what was fantastic about doing this work, because it was also driving these bioinformatics tools like the Lena and the crosstalk algorithm. It was driving it somehow, trying to take out information of your data. So I think that should also not be undervalued, what new information can, push you to do in basic science. And I found that very rewarding.
Magno?
Yes. So now it's my turn.
If you want, yeah.
So I have a few questions. Great talk, Christer, complete basic things, going to most complex biochemistry, and trying to really understand how the system works. So it's a system biology project, I would say. So are you able to see and quantify cholesterol and sterols in general, or how does it work? Does it have any effect when you have this clock of fasting and sleeping and those? Do you have any? Do you quantify that?
Sterol esters, yes, they are quantified, and also the cholesterol. Right, that's what you're asking. Oh, are we losing Magno?
Sorry, I didn't get it.
So we are quantifying correctly, yes. Cholesterol and the cholesterol esters are quantified in the data set, and we also see some oscillations in them, and that is there in the resource data set. And to also discuss that is a little bit, a lot of information, so I spared you that today. Let's leave it like that. But once the data is published, there will be a resource data, so the ones who are interested in sterol metabolism can also go and see that things actually do change, which is quite interesting. Did that answer your question? Or?
So I have another question, is like, how long does it take for a single shotgun analysis?
Depends on how deep you want to go. I can almost say it would be the same in the proteomics, that if you really, really want to go deep, you can also start doing fractionation and things like this, which will then, of course, increase the time on the individual sample. So on a standard basis, what we do per sample, because we're so lucky in the sense that we have the time available, we spend around 1 hour in a routine run, and we do the basic is a 3 × 20-minute analysis with 3 injections per sample of 20 minutes. And if we wanted to, and the people in Dresden, I'm aware, they push it down to, I think, 10 minutes, the same kind of analysis that we do for high throughput and get similar data. So if we wanted to, we could also, reduce the number of repetitions. But my concern is a bit that when you reduce scan repetitions, typically also the data quality goes down a little bit. So I rather somehow use the time to its max capacity to keep data quality high and get good data. So that's why we, by default, do 20 minutes. But as an example, we would now do some flux analysis from data in mice, low-level tracers, where we basically push it up to a 45-minute injection with a nanomate, and then also increase the resolution of the FTMS detection on the FTMS2 to 500,000. And like that, we get beautiful data. And since we are doing shotgun and we have an instrument that can run so long time, we are so fortunate that we can use that time for the sample and then get insightful data. So it depends. Conversely, you can also do 5 minutes if you just want to do screening of some plasma samples from humans. You can even put it down to 1 minute if this is your interest. But the shorter you scan, the worse the data quality typically becomes. So it's a trade-off between depth of analysis and what you need, so to say.
I see. So another question is like, I remember back to 2006, you were trying to map the double bond position of lipids. Do you think it's solved somehow with this your new approach using ozone?
The short answer is
it's a big question in lipidomics, so it's an open question. Let's say.
Yeah, so there's many nuances to that. Is that you can still do cool biochemistry without double bond positions. I hope that I gave some insights to how you can use the technology without knowing where exactly the double bond, and it can still give you relevant, biochemical and physiological information. And then to the more specific analytics is that it will still take us some time until we have this in a routine practice. And in my opinion, I'm a bit surprised why there are not more routine platforms, lipidomics platforms that are having, double bond precision as a standard, analysis. Ozone ID has been out there for more than 10 years. The UVPD and other things have been out there also for 6-7 years in different flavors. To be frank with you, I think it's a sensitivity problem with these kind of, additional ion activation mechanisms. That's one level of it. And I think a lot of these ETD type or UVPD are not specific enough, especially not for shotgun lipidomics. So you have a lot of, unspecificity in the fragmentation patterns. That you don't have to the same level in the OSID is my feeling. That's why I chose to do that. And when you see the UBPD, you have many fragments when you see your fragment spectrum. But that just means that your overall sensitivity drops down because a set of molecules distributes out to many individual fragments. And just by pure Poisson statistics, it gets less sensitive. But in the OSID, you have two main fragments if you have one double bond, and that actually allows you to keep the sensitivity high. This is something to keep in mind when you look at this. Yeah.
I see. And my last question, to give opportunity for other people. So it's just more curiosity. Have you ever tried single cell using shotgun lipidomics? And do you think that you'll be able, how many cells can you see using shotgun lipidomics?
I think for an extraction, we do something like, 1E5 we can use. Like, have I done single cell lipidomics? The answer is no. I haven't really had the interest and the need. And I also think that there are some technical limitations with the single cell analysis in terms of data quality. Mm-hmm. You also see it, in my personal opinion, when you look at even the, the RNA work on single cell level, you only get 4,000 RNAs. compared to an RNA-seq analysis, which goes much deeper, right? So how much is technical variation and how much is actually biological variation in these things? So from my point of view, I'm staying a little bit behind and seeing how the field develops before launching in on that.
Okay. Thanks a lot, Christian.
Thank you, Magno.
We have one more question from Giuseppe. He's asking about the coverage of glycolipid species, like the in ID-wise, using your platform.
So glycosphingolipids in the mammalian system is a very complicated and big, how to say, structural or chemically diverse domain. And I would say if you, from all what is possible to detect out of that big chemical space, we are actually pretty bad. Maybe we are 5%. When you go to liver cells, maybe it's not so important in terms of glycosphingolipids. We do, the main thing we get is GM3, which works pretty well. And then we once in a while we get some GM1, but that's basically it. Of course, we get the hexosylceramides, the diacylceramides. If we go to brain, we mainly have also the GM1 and the GM2, GM2, GM3, and the sulfatides, and then the hexosylceramides. But we haven't invested too much work in getting deeper into the glycosphingolipids. But I guess where there's a will, there's a way. So definitely it could be explored. But you need internal standards. That's then the other challenge. So to do quantification, one thing is whether you just want to do structural elucidation of sugar head groups, or whether you want to do some proper kind of really quantitative work to show differences. And there you need the standards, which are probably not available always. So that's a challenge.
Yeah. Well, okay, I think we can end up here. And again, thank you very much, Christian. It was really nice to see you again.
Thank you very much for inviting me.
Thank you very much for your sharing with us this really, really nice work that you've been doing already for, I would say, 15 years, maybe.
Yeah, time flies when you're having fun.
Time flies, yeah. So thanks again.
Yep.
Thanks everybody to watch us.
Yep, thank you. Have a nice
Thank
day.
you, Chris. Nice to see you.
Yeah, likewise. Hope to see you soon again in lifetime.
Yes.
Sure.
Yep. Ciao.
Ciao.